Assalamualaikum warahmatullahi wabarakatuh. We are from 4 and today we will give presentation about implementation of biological agents to control pests and diseases in corn plant. Before we start, let me introduce our member. First, Noviana Wardani with study number 107. Then Abni Jelang Milani, study number 190. Eksita Kusuma, study number 204, and Abil Milzam, study number 229. Okay, background. According to the Indonesian Central Bureau of Statistics, maize production in Indonesia is low, and as for the DIY region, according to BPS data from the DIY province, corn production decreased in 2015. One of the obstacles faced in corn cultivation activities is the presence of pests and diseases that can reduce corn production. And so far, the control of pests and diseases in corn plant mostly still relies on chemical pesticide, and this is not good for the environment because it can pollute the environment and create resistant pests. And the use of biological agent as a substitute for chemical pesticide needs to be done to minimize the negative impact of the chemical pesticide. Okay, this is what we will discuss today. First, we will discuss about corn plant, and then biological control agents and biological pest and disease control in maize crop. Okay, corn plant. Corn plant is an annual crop. In one life cycle occurs for 80 to 150 days. The first half of the cycle is vegetative growth stage, and the second half is the generative growth stage. Corn is a type of green food crop or cereal from the grass family. Corn is widely known by the Indonesian people because this type plant can be used as a staple food substitute for rice and various kinds of processed food. In addition, part of the corn plant can also be used as animal feed such leaves, stem, husk, and cob. Corn plants grow in the lowlands to hike up to 1,200 meters above sea level, require clay soil media, sandy loam, volcanic soil, fertile, loose, rich in organic matters, requires at least 8 hours of sunlight per day, air temperatures 20 to 33 degrees Celsius, rainfall medium, soil pH 5.5 to 7 with good drainage.
Biological control agents. Biological control is a natural phenomenon that consists of regulating the number of species by natural enemies, which constitute the agent of biotic mortality. Thus, all living beings have natural enemies attacking their various stages of life. Among these, there are quite diversified groups such as insects, viruses, fungi, bacteria, nematodes, mites, and spiders. Abil et al. 2015. Nowadays, the biological control is indispensable in the integrated pest management program because it combines cultural and chemical practice for resistant management with real gains in grain yields. This tool is the way that rationalize the control of pest insect of crops with natural process and the rational use of agricultural pesticide for pest control. Simonato et al. 2014. Biological pest and disease control in maize crop. The first one are corn stem borer. This pest attacks all parts of the corn plants, and in heavy attacks the yield loss can reach 80%. Newly hatched larvae are yellowish-white, feed on the move. Young larvae eat the tips of male flowers or panicles and after advanced instar will burrow corn stalks. Symptoms of this pest attack in the form of small holes in the leaves, slit in the stem, malflowers or the base of the cup so that the stem and tassel are easily broken. One of the most commonly used biological control for the control of Lepidoptera in maize is Trichogramma sp. The female individual of this parasitoid lays her eggs inside the egg of the plague, and the end of the biological cycle emerges the adult. The great differential in favor of the exclusive biological control again of eggs consists in the reduction of the population of the pest, preventing injury to the plant, being efficient in the pest control Spodoptera frugiperda, Helicobacter, Helicoverpa armigera, Diatreae saccharalis, Elasmopalpus lichenocelus. In the parasitism of the eggs of Agrotis epsilon, Trichogramma pretizoum, and Trichogramma atopovirilia stand out, demonstrating high potential for use in pest management. The main strategy used to combat Spodoptera frugiperda is the use of corn hybrids, which express a type of insecticide protein called Bt. Junha 2017. This bacterium is effectively toxic to the insect of the order Coleoptera and Lepidoptera, being an alternative of control aiming the management of pest insect is the maize crop. Biological control with Bacillus thuringiensis has shown good results in the control of Spodoptera frugiperda. The next one is armyworm caused by Spodoptera frugiperda. The larva damage plant tissue by feeding with chewing mouth parts. The potential for feeding black cutworm larvae to kill plants, thereby reducing stand and potentially yield, makes large infestations of black cutworm a serious threat to corn and other crops. Larvae are active mainly at night. Small larvae feed on leaves, creating irregular holes, and can cut small weed seedlings while feeding near or below the soil surface. Fourth instar and larger larvae can cut off corn plants, sometimes dragging the cut plants below ground. Plants cut above the shoot apical meristem or growing point usually recover. The next is Bevaria bassiana can suppress a epsilon third instar larva. Mortality percentage increase with increasing concentrations and time elapsed after all treatments. In spite of the slow action of Bevaria bassiana, it revealed high mortality with LC50 66.73 x 102 conidia per milliliter and LC90 1003.704 x 103 conidia per milliliter. Both Tritis and Metarhizium fungi are also parasitic on larvae and cause high mortality in Java and Sumatra. The next is slate wilt disease caused by Cephalosporium mitis. The primary disease symptom is a rapid wilting of maize plants, generally 70 to 80 days before tasseling and until near ripeness. The pathogen can cause damage to sprouting seeds, which result in reduced and delayed seedling emergence. Necrotic lesions may appear on the roots of susceptible maize plants as early as 3 weeks after inoculation, and at that time, infected seedlings have shortened roots. First above ground symptoms usually appear approximately 60 days after sowing. Biological control of plant pathogens by microorganisms has been considered a more natural and environmentally acceptable alternative to chemical treatment methods. These microorganisms that display antagonistic effect against target pathogens are capable of survival and propagation in target location. In agriculture, the use of bacteria and fungi antagonistic towards plant pathogenic fungi has been suggested as an alternative to chemical pesticides for control of plant disease. Therefore, a wide range of different microorganisms was used to suppress lead wild pathogen of maize. Trichoderma herzianum and Bacillus subtilis were successfully used for biological control of cephalosporium wilt disease of maize.
Okay, the next is Fusarium corporate disease caused by Fusarium verticilioides. Biological agents that are effective in controlling Fusarium include other are Bacillus ameloequivalens and Bacillus mojavensis. The two biological agents can improve seed quality and reduce fumonisin toxin content due to Fusarium infection. The treatment of Pseudomonas bacteria formulated in the form of a seed treatment and applied drug spraying can reduce Fusarium infection and suppress fumonisin toxin production. Pseudomonas found in the root area of corn plants and can function double, which products growth regulators and corn plants can function double. Which product growth regulators and act as a solvent phosphate. Bahrudin et al. Suggest that fluorines can produce cyanida, which acts a toxin as blocking pathogen development. Meanwhile, Cavaglia et al. Report that the bacterium Azotobacter armeniaceus was effective in reducing Fusarium in the root area of corn plant. Okay, the next is Anthracnosa caused by Caltotrichum graminicola. Anthracnosis symptoms vary widely depending on numerous factors such as a genotype, age of plant, and environmental condition. The symptoms is like a small over or elongated water soaked spots which enlarge up to 15 millimeters long appear on leaf. Next symptom is like a lesion develop a tan center and red, brown, or orange border. The next is lesion mycoses to form a large necrotic dead patch. The next severely infected leaf unsusceptible hybrids may wither and die. The next symptom is like a fungal fruiting bodies develop on dead tissues and may produce pink or orange spore masses. Okay, the next is to control anthracnosis caused by Calyptotrichum. It's like a corn leaf anthracnosis caused by Calyptotrichum graminicola can be controlled by seed treatment with Trichoderma hergenium. The result implies that Trichoderma hergenium can systemically induce concealing defense response to pathogen infection. Further observation shows that the activity of specific cytolytic enzyme is higher in plants grown from Trichoderma sp. treat seeds than in control plants. Okay, next. Okay, this is the last of conclusion. One of the obstacles faced in corn cultivation activities is the presence of pests and diseases that can reduce corn production. Some of the pests that attack corn plantation is corn stem borer, armyworm, cutworm. Then some diseases that attack corn plantation is like a lateral disease, Fusarium corporate, and anthracnose. The pests and disease can be managed with biological agents. Okay, thank you. That is the presentation. Any question? Wassalamualaikum warahmatullahi wabarakatuh.